Welcome to Let's Talk Micro. Hello, everyone. Welcome to another episode of Let's Talk Micro. I hope you had a great week and I hope you enjoyed last week's episode on opticin and microdase disk. So, before we get started, remember that Let's Talk Micro is available on Apple Podcasts, Spotify, Google Podcasts, Stitcher, Audible, Amazon Music, TuneIn, Pandora, Overcast. So, whatever you listen to your podcast, you can listen to Let's Talk Micro. It is also available on iHeartRadio. So, you just search Let's Talk Micro and you'll find me there. On some of the sites, you might have to scroll a little more than others, but you can see the picture. I am also on Instagram as Let's Talk Micro and on Twitter as Let's Talk Micro 1. So, please go ahead and follow me. I like to post pictures of organisms, of the biochemicals that I'm talking about. So please go ahead and follow me. I can't believe we're already on episode 9. So we're really moving along. There's so many topics to talk about. So I want to appreciate all of you that have downloaded the podcast and listened to it. All of you students out there, all of you microbiologists out there, go ahead and share it. Tell someone about Let's Talk Micro. I'm trying to promote it as much as I can through social media. So whenever you have your fellow students, co-workers say, hey, I listened to this podcast about micro. It's interesting. It's basic micro. So go ahead and spread the word, please. I truly appreciate it. So on our last episode, I finished going over the biochemicals for gram-positive cocci and rods. For your GPRs or gram-positive rods. Mostly you use your catalase. There are more out there, of course, but your very basic one is the catalase. And some species, of course, are positive. Some are negative. And then the gram-positive cocci, we talked about catalase, coagulase, PYR, strep typing, opticin, and microdase disk. With these biochemicals, like I mentioned in the last episode, you'll have no trouble performing some basic IDs on these organisms. Unfortunately, as we move along, like we get more modern with more technology, with the Molotov, some of these biochemicals, they are being used less and less. They're very helpful, like I mentioned before. And I think I've seen this mostly on the younger generations, no offense, of course. But I can see the reasoning. It's like, why perform all this stuff when I can have an ID, in 10 minutes? I do get it, but like I always say, and once I talk about automation, I will build up more on this. But you have to know what you're looking at. You have to recognize the morphology of your organisms. You have to recognize the biochemicals. So you know that you have a correct ID. Because, right, the Molotov does not set itself up. It takes a person to actually set up the sample. It takes a person to scan the ID. So there's human error here. So you can scan the wrong patient if you don't know what you're looking at and you're not the one that it's performing the Molotov yourself, because it happens, especially if you're a large facility, you have so many cultures to go through that you might have a person that's assigned to doing Bitech or doing Molotov. So what if this person doesn't know what they're talking about? What they're looking at. And you tell them, go ahead and, you give them a plate. You write on your plate, moldy. And then they don't know the organism that they're supposed to set up. They set up the wrong one. And then the person that's accepting the ID might be you, or you might have some help. Maybe that person doesn't know what they're looking at either. So they accept the wrong ID. So then you release that wrong ID and the patient might get put on the wrong treatment. So this is something that I always like to say. even though, and don't get me wrong, I have nothing against the Molotov. I love it. It makes your life easier in so many ways. For example, when you're working anaerobes, you release that ID so fast, and you're done with them. When you're trying to ID organisms that do not require susceptibilities, like Erococcus urinae, you put it on the Molotov, you ID it, you're done. With your group A, group B strepes, it makes it easier. Staph saprophyticus, you typically don't do susceptibilities on the urine bench. You see it, it fits the criteria, you want to rule out Staph saprophyticus, you put it on your Molotov, and then you're done with it. So it definitely makes your life easier in a lot of ways. But however, it causes that problem that I've been talking about, that you want to, it's going to give you the answer so you don't do the biochemicals. And that can create some problems. And you can potentially put your patients in harm. And this is all about the patients. So, and it's interesting because I was working in the lab recently, and the Molotov was down because there were no supplies. So now we're doing back everything old school. With your doing as many biochemicals as you can, doing IDs. So some techs are saying, "Oh, I don't like this. It seems it takes longer." So I don't know. I mean, I think it's a matter of adjusting your process because people did not like the moldy at the beginning. And I think sometimes, it can take you a little bit longer, especially if you're in line waiting to put your slide in. And don't worry about it. I will touch on the Molotov on a later episode. But at least you microbiologists that work in the lab, you know what I'm talking about. So I think sometimes it might take you longer. But we'll talk about that later. And I always like to touch briefly on, maybe sometimes not so briefly, about what I covered in the last episode. So we talked about, right? We talked about Optokin, which is not a biochemical, but it's a helpful tool as part of your differential. Streptococcus pneumoniae is a serious pathogen, and we need to rule it out. So typically it is mucoid, and you need that zone of 14 millimeters to presumptively identify it as strep pneumo, right? If the organism is susceptible to it, there's a ring around the disc, and you measure that zone. Sometimes you don't get that zone, and you need to perform an alternate method of ID, such as Vitek, or like I mentioned, the Molotov, and you get that ID fairly quickly. We also talk about the microdase disc. You see micrococcus mostly in blood cultures as a contaminant, right? We talked about that. And this is, this disc is a quick method of identifying it. So, typical presentation, you have a positive blood culture, gram stain is gram-positive cocci in clusters. You put a sample in your nucleic acid method, like your Luminex. It comes back as not detected. It doesn't detect an ID, doesn't provide you an ID. And then on the bench, you get micrococcus. So you go ahead and put some colonies in that disc, observe it for your color change, and you're done. And then you go ahead and release it as micrococcus species. And that's the end of it. So we have officially wrapped the biochemicals for the gram-positive cocci, and we touch on the rods. Always keep in mind, there are more, but these are very commonly used nowadays. I mean, especially in the US. I want to start doing maybe a segment where I talk to techs from other parts of the world to see what they're doing, and compare and contrast. But we'll get there. But there are more biochemicals out there than these, of course. But with the ones that I have talked about, you'll be good at identifying most of the cocci. So today we start talking about biochemicals for your gram-negative rods. There are also many out there, and two of the most basic ones are indole and oxidase. So let's go ahead and start with indole. So what is indole used for? It is used to determine the organism's ability to hydrolyze tryptophan to form the compound indole. So a color compound is formed, and you have different indole tests out there. One of the most common ones used, it's the DMACA, which is made by BD. With this one, a positive reaction is a blue color, and a negative reaction is when you have no color change. Right? So positive is blue, negative, there's no color change. So which gram-negative rods do we use this for? Well, of course, one of the most common groups that we use the indole for, most common type of gram-negative rods, it's the Enterobacteriaceae. And what are the Enterobacteriaceae? Well, this is a large group of gram-negative rods, and we're gonna get kinda technical. So some of its members are part of the human intestinal flora. They are facultative anaerobes, and these are actually some of the most common organisms encountered in the clinical lab. So, like I said, some of it are part of the human intestinal flora, like E. coli, Proteus, Morganella, Klebsiella, and some are not. Can you guess which ones aren't? Well, let's start with Salmonella and Shigella. All of you have heard about Salmonella out there. When we get these outbreaks, like in the peanut butter, and some eggs. So definitely Salmonella is part of this group, but it is not part of the human intestinal flora. It is part of the animal flora. Which brings me, I always like to tell stories of things I heard that I have been through. So I met this tech once that she worked for her hospital had a contract with a zoo. So they brought samples over there. So she saw all kinds of samples. Like she tested cultures on camels, snakes, chickens. She said it was kind of like a little freaky when you do like a stool culture on a chicken, because as you know, salmonella is part of the chicken flora. So you get all this, this plate full of salmonella, and you're like, oh my god, and then you're like, wait, it's chicken. I don't know. I find it humorous. But of course, in humans, salmonella is not part of the flora, so it is pathogenic. And definitely the states, depending on the state that you are, all the states, it's like a reportable organism. So when you have a salmonella, you have to send a sample to the state you work on so they can do the proper typing. That way they keep track for epidemiology purposes to make sure that you have a case, it's either like an isolated case or you might have some sort of outbreak. So that's why, of course, since it's part of the chicken flora, you have always, make sure that you cook your eggs properly. When you have like cookie batter, it tells you it has like raw egg. Make sure you heat your product to the appropriate temperature. Always with your chicken, make sure that whatever you're preparing your chicken, always disinfect your area. With your bleach wipes, properly clean the area. That way you don't get sick. It can cause gastrointestinal problems, diarrhea. So it's a serious organism. So that's why as soon as they detect an outbreak, they announce it on the news and they let us know and they pull the product. They do a recall on it. And then we have Shigella, which is also not a part of the human intestinal flora. It is only found during time of infection. So, these organisms, they're typically transmitted, like I said, with salmonella when you have contaminated products. Um, and also when you have overcrowded areas, so it can be transmitted from person to person, spread by the fecal oral route. So, you know, they have been a part where there's human waste, like stool, and maybe the area is not properly clean. When you don't have clean water, you might get contaminated. You might get sick that way. So these are two very serious organisms that are part of the Enterobacteriaceae. But going back to the Enterobacteriaceae as a group, so the other ones that I mentioned, they can be their intestinal flora, but then they're pathogenic outside other areas of the body. Like, of course, E. coli. E. coli, some strains of E. coli can cause some serious GI problems, but overall, it's intestinal flora. But when you have it outside of that area, it can cause problems. I mean, E. coli, it's the number one cause of UTIs. It's the most common one. So if you're on the urine bench, you see E. coli once, you see it 40, 50 times. It's a very common organism. So with the Enterobacteriaceae, the indole is an important test. So you have on this group, some species are indole positive and some species are indole negative. And let's talk about that. So you have, for your positive, let's start with the positive. So you have E. coli. You can have Proteus vulgaris. We can have Klebsiella oxytoca. Now, granted, there is a small percentage of E. coli that can be indole negative, but overall they're positive. So when you have an indole negative organism that it is identified as E. coli, you have to confirm that. Repeat your indole and confirm that ID by another method. One fact about E. coli is actually it's very similar at the RNA level to Shigella. So some instruments like the MALDI-TOF, they cannot differentiate between both of them. So at that point in time, you do your test. I mean, E. coli is indole positive, and that should be the one that will differentiate it. And of course, if it's lactose fermenting, that's another one. But E. coli can be also non-fermenter, and that's something that we'll talk about another time. And then Proteus vulgaris, like I said, and Klebsiella oxytoca. And then we have some that are negative, like you have Proteus mirabilis. You can have Klebsiella pneumoniae, Enterobacter cloacae, and Enterobacter aerogenes, which is now called Klebsiella aerogenes. And I've been saying all this time Enterobacteriaceae, that's when we started. When I started, that's what they were called. Now they are called the Enterobacteriales. And for you students out there, these are very typical questions when you're doing your clinical rotations. I have mentioned also that I dab in teaching, and I like to ask these questions as well. If you have a Klebsiella that's indole positive, what's the species? And then you will say Klebsiella oxytoca. And then if it's indole negative, Klebsiella pneumoniae. So these are questions that we like to do case studies, or we give you, let's say I give you a plate that has Klebsiella, and I say it on the question, this organism was ID as Klebsiella. The tech performed an indole test and it was negative. What is the most likely ID of this organism? And that's what you say, Klebsiella pneumoniae. Or if you're given the same question with an indole positive result, that's where you say, well, it's probably Klebsiella oxytoca. The same thing with Proteus. That's another question that techs that are training you, lab instructors, they like to ask. If you have a Proteus that's indole negative, what's the most likely ID? versus indole positive. So you will say for negative, Proteus mirabilis, and then for positive, Indole vulgaris. And that's at the basic level when you're learning. I mean, once you get to the bench, you know that there's also Proteus, there used to be the Proteus vulgaris group, where you have Proteus vulgaris, Proteus hauseri, and Proteus penneri. But now we know that Proteus penneri is indole negative, and then hauseri and vulgaris are positive. But when you're learning, when you're getting started, there's so much information. I mean, this not only applies for micro, but for other areas as well. So we kind of like to keep it simple when we're teaching or we're asking you on the ASCP. And then as you get to the work area, you start learning more and more. So for your indole positive, Proteus vulgaris. For your indole negative, Proteus mirabilis. And then also, now we start moving away from the Enterobacteriaceae or Enterobacteriales. And then we can also use your indole for your Pasteurella, which is positive. And then Haemophilus, which is positive as well. We can also use it for non-fermenting gram-negative rods. And what are non-fermenting gram-negative rods? They are a group of organisms that they do not ferment lactose. They are implicated in various diseases such as UTIs, septicemia, and some of these are actually seen in the cystic fibrosis patients. Some of these they include, but they are not limited to Pseudomonas, Burkholderia, Acinetobacter, Stenotrophomonas, Echromobacter, Aeromonas, Flavobacterium, Myroides, Comomonas, and Shewanella. And a quick story about Shewanella. Well, it's not a story. It's just like I encountered it a few years ago. So Shewanella is normally it's an environmental and food organism. It is not part of the human flora, and a lot of times it's transmitted when the infections they're associated when the person is exposed to aquatic and marine habitats. And you typically see it in mixed cultures, and it has been implicated in cellulitis, otitis media, and septicemia. So I definitely see it. I have seen it mostly in some mixed cultures. A lot of times you have diabetic patients, they have these wounds that they would not heal, and you have at least 3 or 4 organisms. You have a combination of Pseudomonas, Stenotrophomonas. Sometimes you have Acinetobacter. In addition, you have many Enterobacteriaceae. So it's a lot of organisms. So at that point in time, you have to make a decision based on the guidelines and what you work up and what you mix. But circling back to Shewanella, it smells horrible. And it's not like you're actively, I always say, you should not smell plates in the lab. So I'm not actively trying to smell it, but it smells like, I don't know, I have like something is decayed, like something dead. So I don't know, maybe like some really bad trash. I have never smelled like a decomposing body, and I'm glad and I hope I never get to. But I will think that that's what it will smell like. It was such a foul smell. But yeah, there's Shewanella for you. As a micro nerd, it's always exciting when you see these organisms that you rarely see, at least in my facility. You might work in a facility where it might be more common. But like I said, it's just I tend to see it more in those mixed infections, those mixed cultures. And so as far as results for your indole on your non-fermenting gram-negative rods, we also get some varied results. You have Pseudomonas aeruginosa, which is your most common non-fermenting gram-negative rod that you see in the lab. So for your Enterobacteriales, you see E. coli, and then Pseudomonas for your non-fermenters. So just like E. coli, you will see it in almost every source that you get, like any wound culture, urine culture, respiratory cultures. So there's very common organisms that you can see them outside, where you can see them in many parts of the body when you're working in the bench. And then you also have Aeromonas, which is positive. Aeromonas is a stool pathogen, and it causes infections. It causes gastrointestinal infections. And then you have Flavobacterium, which is Indole positive. Other two that you have are Acinetobacter, which is Indole negative, and Stenotrophomonas, which is negative. So you get some mixed results with your Indole here. But definitely, this is a very important test. It's always good to know expected result. That way, when you get that ID, you can correlate it. You know what you're looking at. If you get a questionable reaction, you correlate that with your organism. And that way, you make sure that you are releasing the ID of the proper organism, so the patient can get the appropriate antibiotic therapy. And that brings us to oxidase, which we will actually talk about it on the next episode. And that, my dear audience, is the end of this episode. I hope you enjoyed listening about Indole. Like always, I enjoyed talking about it. Stay motivated. Stay safe. Continue bringing that motivation to your job. Do the best you can. When yesterday I was in the lab, and I started my day with a huge stack of plates. There's no better start to your day than that. So continue your motivation, continue bringing your passion, and continue talking micro. I hope you enjoy the rest of your week, and until next time.